So I'll go ahead and begin. Hopefully it opens and everything. If it opened, can someone give me a thumbs up? Is it not opening? Crap. What does it say?
Mine says it's not going to open till 5:20.
Okay.
But it's 5:04 on my computer.
Yeah, it's 5:04 on my computer too.
No. What the heck is wrong with my computer? Okay, let's see. For Lab 08.
Mine's available
Mine's
now.
open.
Okay.
Yeah,
Cool.
mine opened up.
Cool. Awesome. Yay. Good luck, everyone. Let me know if you have like a question, and you can also type in the chat individually to me if you have like an individual question. Sorry to interrupt y'all. I have a few questions about when you're done. If we were in lab, I'd just make y'all sit there and like practice meditating. So do that now. Just like breathe in, breathe out, breathe in, breathe out. It's really relaxing. Right, we got 5 minutes left, and I think we have 2 more people working on it. So everyone just chill. Okay, cool. I think everyone has finished it now. Anyone still working, definitely unmute me and let me know. Cool. All right. So hopefully it wasn't too bad. Normally I'd go over it right now just to walk us through it, make sure we understand. But I think I have a few people that it didn't open for, so I had to extend it for them. So hopefully next week it'll work for everyone and we'll be able to discuss it afterwards. But if you see it's kind of, application based, I don't like just, fill in the blanks or anything. I like, real life pictures and stuff like that. So that's kind of how the quizzes will be for the rest of the semester. So I have a few announcements that I want to make. First of all, I'll be doing attendance based off of the Zoom report. So make sure you don't leave early because I'll be checking the time that you checked in and checked out. So if you check out early, you won't get attendance for the day and I'll have to dismiss your quiz grade, put it as a 0. So definitely don't leave early. If you ever do have an event that, you do need to leave early for, like if you have a doctor's appointment or whatever, email me and we'll figure it out. But again, if you weren't here last week, we never will ever go the full 3 hours. We normally will finish by like 6:30. So it shouldn't be that bad to stay on. So attendance, I'll be doing that after lab. Make sure you stay the whole time. Just another announcement for the quiz, in case you weren't here last week. I'm not going to be very strict about it this week, but it is closed note, so make sure that you are not using your notes. I'm watching y'all over the camera, but if you go to a Quizlet or anything like that, I get the notification on my screen. So I'm gonna let it go this week, but next week definitely make sure you're not switching to a different website. Make sure you're staying on the Canvas page until you finish up. I can also see when you're done. So I did not find that setting until while we were taking the quiz today. So once you submit it, it lets me know. So if you submit it and you want to do something else on your computer, just send me a message in the chat. So let's see. I had some emails that I'm so sorry to get back to you. Just this week has been chaotic. My family evacuated for the hurricane. We didn't get damaged, thankfully, but I've been super busy this weekend trying to catch up from the schoolwork I didn't do last week. So I'm going to address those now. I got an email from Alexis about the syllabus, the schedule, and the readings. They were not correct until last night. So your question is totally a good question. So let me pull up the Canvas. I'm going to walk y'all through this really quick one more time with the new edits that I made. So I had to combine our lab with lab 16 because that's the other lab I teach. So I'll discuss what happened there afterwards. I had some emails about that, but the Canvas page is going to look a little different now. We have that little introduction. If you see here, y'all are Lab 08. You definitely want to remember y'all are L08. Because there will be multiple things on Canvas that will say, Zoom for L08 versus Zoom for L16. So definitely keep in mind your L08. If you ever email me, try and let me know that you're in L08 too, just so I can keep in mind who's in who. So y'all are Lab 08. I have everything for L08 highlighted in yellow, but I made this schedule and the yellow is kind of hard to see, so I might change y'all's color to like red or green or something. So here's your schedule that I made. Y'all are the yellow ones, lab 16 is the green ones, and this is all your due dates. So lab report 1 was due last night. You can see that there. We have our quiz today. And so this is just kind of a way, if you ever think, oh, do we have something due? Just go check this and I'll update it month by month. So you can always have your due dates in front of you. We don't have any exams or anything this month, so it's just lab reports and quizzes. We will have a cat dissection at the last week of this month. If you see next week, we don't have labs, so you don't have to worry about anything. Your next lab report isn't due for 2 weeks. Your next quiz isn't for 2 weeks, so y'all just take it easy, Labor Day week. So there's that. I did update the syllabus. It did say like LO1 through LO5. It only said that because it wasn't updated from the summer syllabus, but it is for all labs in case you saw that. You can also click here for a downloadable version of it, and it looks kind of nicer. If you scroll down here, this is so complicated. I absolutely hate this, but I had to just copy the syllabus that the other IAs are using. There's a lot of complicated things in here that the other IAs are going to do that I'm not, because it's on Zoom and this semester is already hard enough. So just ignore this at the bottom and just go off what I have here and on the modules. For your readings, I did not post them on here. I might redo that, but I did have some questions on what y'all should be reading, and where you can find that is on the module. So here's today's module. I've updated most of the modules. So where you can see is the readings for the lab are going to be posted in the module. And I guess I'll start posting under your assignments, like read this for next week. The readings, you don't have to like heavily read them. Skim through them. They have really good notes. Where's my manual? I need to find that. So when I do my readings, I just kind of like highlight things and stuff like that. But if you're not a reading person, just kind of skim through it. But if you like to read, here are where you can find the readings. So I had a few questions about the lab report in this discussion from last week. When I combined our labs, a lot of stuff got deleted on accident. So if yours like got deleted or whatever, I might just give everyone like a 100 for that, because it's going to be complicated. So if I see that you didn't turn it in, I'll just waive that for you. So this first week's kind of a pass. I'll get it. There won't be any deleting things going on next week. Hang on. Come here. Stop. Oh, my cats are tearing stuff up in my room. Okay. So yeah, if there was any error there, I'm just going to go ahead and waive it for this week. Hopefully the lab report wasn't that bad. Again, this was like kind of a boring one. They'll get more exciting as we get more into the semester. So those are my announcements for today. Again, we don't have lab next week, so you won't have anything due for 2 weeks, but definitely keep in mind when we get back that we will have things due. So frustratingly enough, I forgot to do these 2 chapters from last week. Normally they have their own lab, but I guess because of Labor Day, we are going to combine them all into one. So we're technically having this lab and this lab today, but I'm going to zoom through these first 2 because you already learned these in biology. I don't put these on the practical very much because I don't like biology. I like anatomy and I like the human body. I don't like the cellular level. So we're just going to get through these 2 really quickly. If you want to know more about it, I am more than happy to make a Zoom appointment with you where we can discuss the details. But today, I'm just going to get through what I want y'all to know for these 2. Then we'll get into the tissues and skin, and that will be a little more in depth. So our objectives, we just took the quiz, we're going to do the labs, and then if we have any questions, whatever, after lab, talk to me. So here are the 2 PowerPoints we're going to go over today. If you want to open them, you can. I'd have your notes ready, or however you're taking notes. If you want to do an outline on your computer while we do this, definitely feel free to. And again, if you have any questions during the lecture, feel free to interrupt me. So let me get our PowerPoint pulled up. So if we want to follow along, should we be looking at the lab portion or the readings? I would definitely be looking at the PowerPoints I post. So I have made all of these PowerPoints on my own, and I make them directly from the lab manual, but I condensed it into what I think is important. So I would definitely take notes off of the PowerPoints. You'll never see a question or an exam question that's from the readings. It'll all be from the PowerPoints.
Gotcha. Thank you.
I'm not like technically allowed to tell y'all that because you might be like, oh, I'm not gonna get the manual. But that's how it's gonna be. So yeah. Okay, cool. Anyone else? Alrighty, again, interrupt me. I like being interrupted. So, okay, so it's my screen sharing. 2, and let me get it into full screen. Can y'all all see the PowerPoint? So give me like a head. Okay, cool. So this is chapter 2, cells and mitosis. Again, kind of just a review from biology. So our objectives are going to be looking at prokaryotic and eukaryotic cells. We're going to look at the major organelles. We're going to look at the phases of mitosis. We'll look at the DNA molecule and how it's arranged. I think we're probably going to skip over that. So we have 2 types of cells. We have prokaryotic cells and eukaryotic cells. So you've probably heard of prokaryotes and eukaryotes. Prokaryotes have no nucleus, so the nucleus is kind of known as the control center of the cell. So they are just kind of like really basic organisms like bacteria. So they don't have much importance. Then we have eukaryotes. So these have a defined nucleus. You can see that right there. So these are going to be like our plants, animals, humans. Y'all don't have to study that, just a distinguishment to make. So now we're going to look at the cell structure. So we all know that the cell is surrounded by a membrane called the plasma membrane. It, contains all of the contents in the cell. Am I recording? Let me make sure. Yes, and I forgot to tell y'all when I was on Canvas where to find the Zoom recordings. I'll do that at the end of lab. But that plasma membrane just keeps everything inside the cell. It's the barrier to the outside, so our cells are just floating around in our body and in different places, and it's just keeping the cell contained and not just floating around. So it has that lipid bilayer that we all know about, the one that kind of looks like this. We all know what the lipid, and I think it actually looks more like that with the 2 sides. So that's that lipid bilayer, and what that does is allow transport into and out of the cell. So we have passive transport, which requires no energy, and active, which requires energy, but we get to that later in the slides. But yeah, so that plasma membrane, lipid bilayer surrounds the cell. Easy. Then we have the cytoplasm. So inside the cytoplasm, which is this whole just space in here, we have the cytosol, which is that kind of gel, the fluid that's inside the cell that will surround the organelles. So whenever you get into exercise physiology, resistance training, and conditioning, all of those, the cytosol is actually very important. It's where we get our energy from in the first 2 to 3 minutes of exercise. You don't need to know this for our class, but just to kind of prepare you, the cytosol is where we produce ATP for the first 2 to 3 minutes of exercise. Again, y'all don't need to know this, just an idea. So ATP is our energy source. It is how we function, how we move, and things like that. So that's I just broke my pen. That's how we get our energy in the cytosol. And then next we have the organelles. So these are the organelles, you know, the nucleus, the mitochondria, all of those guys. So let's look at those more in depth. So first we have the nucleus. We all know from, you know, earlier schooling, it is the control center of the cell. And why this is because it contains genetic material, AKA DNA. So the nucleus controls what goes on in the cell due to DNA. It has its own membrane. It also has a little thing called the nucleolus and chromatin and DNA, but you don't need to know any of these three. So just know that the nucleus is the control center and contains DNA. Next, we have the endoplasmic reticulum, the ER. We have two types, the rough ER and the smooth ER. The rough ER contains ribosomes and the smooth ER contains no ribosomes. You know, again, the function of rough ER is converting mRNA to protein. Smooth ER is synthesizing lipids. I'm a senior ESS major and I've never had to deal with endoplasmic reticulum in any of my classes, so you don't need to know about it. Same for Golgi apparatus. You'll look at the Golgi tendon organs, which are these little guys that detect length and things like that, but you never really look at the apparatus. What it does is just package protein for, you know, transport in and out around the cell, but you don't need to know that for my class. I do want you to know the mitochondria. If you remember anything from public school biology, it is that the mitochondria is the powerhouse of the cell. I don't know why they emphasized that so much for us, but they did. So this is where we turn nutrients, which, by the way, if you ever hear the term nutrients, we are talking about carbohydrates, proteins, carbohydrates, proteins, fats, and what am I forgetting? Yeah, those are the three nutrients. The mitochondria is just turning those nutrients into ATP or energy. So after that first 2 to 3 minutes of exercise, and again, this is just a future thing you'll learn, you don't need to know it now. After those first 2 to 3 minutes of exercise, we'll start taking energy from the mitochondria, which is where that energy is now being produced. So definitely an important part of the cell. As you get stronger, as you build more muscles, you'll start to see a lot more mitochondria inside the cell. As that mitochondria keeps reproducing, getting more and more, that's whenever you'll start to see, you know, muscle growth because we're enlarging our cells, things like that. Then we have lysosomes, which break down waste. Don't need to know that. Cytoskeleton will maintain the cell structure and allow some cell movement. Don't need to know that. Any questions? And I'll always do that little countdown because I always feel awkward whenever my professor is like, any questions on Zoom? And then you're like, do I ask or are they going to start talking? And then there will be like an awkward, like we both start talking at the same time. So I'll always do like a little countdown. It might annoy y'all, but I just don't want anyone to not ask a question because they're scared.
Is this something like more for review, or is it something that we're going to hit really hard?
So definitely this is just a review. At the end of our Zoom today, I'll go tell you exactly which ones I want you to know,
Gotcha.
which I think is just the nucleus and mitochondria. But I'll definitely let y'all know.
Okay.
Cool, cool. Anyone else? All right. Oh, do I? Okay. All right, so we've all heard about the cell cycle. We have interphase and mitosis. I'm definitely more concerned with mitosis than interphase. I don't remember interphase. I don't super understand it. There's that G1 phase where the cell will grow. Then the S phase where it will replicate, and then the G2 phase where it completes growth. I don't remember that, so y'all don't need to know it. You won't ever see that unless you're like a organic chemistry major or something, or biology. But if you're biology, I don't think you should be in this lab because this is a non-science major lab, which makes me super mad that ESS is not considered a science major, because we do have to take really hard science classes and we should get that credit, but for some reason we don't. But whatever, our lab's a lot, or our AMP is a lot easier than the science major AMP. So interphase, don't need to know that, but we all know the saying for mitosis, PMAT. Prophase, metaphase, anaphase, telophase, PMAT. I want y'all to know that. So this is looking at the cell cycle. We have that interphase, don't need to know. Here's prophase. The two, what are those things called? I think I have it listed somewhere. I think those things are called chromatin or something. Does anyone know? No, I can't remember. I think they're called, chromosomes. Okay, the chromosomes. So the chromosomes in prophase are starting to pull apart. Metaphase, they're starting to come together. Remember, metaphase, middle. Metaphase, middle. Know that. Metaphase is the middle whenever those chromosomes come together in the middle. Anaphase is whenever they start to pull apart again, and then cytokinesis is when they become their own cell. So it goes prophase, metaphase, anaphase, telophase, cytokinesis. I just want you to know the order: prophase, metaphase, anaphase, telophase. And again, there's that saying PMAT, P-M-A-T, prophase, metaphase, anaphase, telophase. And I might have a question like, what phase is it whenever the chromosomes align in the middle of the cell? Just because metaphase middle is easy to remember. Okay. Then we have DNA. DNA's name is deoxyribonucleic acid. What it is a sugar molecule that has 5 sides. If you break down the name, D means without, oxy means oxygen, and ribose means sugar. So sugar without oxygen. It has that double helix structure. So like the ladder when it uncoils, and those ladder sides are guanine, cytosine, adenine, and thymine. Y'all don't need to know anything on this slide. I haven't seen it ever again in any of my ESS classes in the future. So y'all don't need to study this. I'm not gonna ever ask y'all anything about this. Any questions? Righty. So now we're gonna get into chapter 3, which is diffusion and permeability. So far we've done chapter 1, we've done chapter 2, now we're on chapter 3. So what chapter 3 is looking at is distinguishing between simple diffusion and osmosis. We're gonna look at solutes, solvents, and solutions. We're gonna look at hypotonic, isotonic, and hypertonic solutions, and then we're gonna look at osmotic pressures. Again, these objectives might stress you out. It's really not that bad. So diffusion, do I have a video for it? Let's see. How do I get it? So diffusion is the passive transport of molecules from an area of high concentration to low concentration. So that's another thing that's probably drilled into your mind. Molecules move from hot areas of high concentration to areas of low concentration. Whenever you see the word passive, that means it does not require ATP energy. It does not require any energy. If you're not familiar with ATP being energy, definitely understand that anytime you see the abbreviation ATP, which the whole word is adenosine triphosphate, which you don't need to know that yet. You'll learn that in your lecture at some point. But anytime you see the word ATP, it means energy. They're used interchangeably. So definitely associate energy with ATP and ATP with energy. So again, passive transport does not require energy. So diffusion and osmosis always kind of confused me for some reason. So I found this video that we'll watch. I think it's really short, but it kind of just explains what
Welcome to
diffusion
Moving
is.
Math and Science. In this video, I'd like to show some examples of diffusion. Diffusion is when objects move from an area of high concentration to an area of low concentration. Check out this example of diffusion. You can see another simple example of diffusion when you watch the food coloring diffuse into the water. If you take an egg, soak it in vinegar, and then place it in water, the water will diffuse into the egg, causing it to get larger. Place sugar into coffee, it diffuses out into the coffee. Watering plants diffuses the water into them. When you go into Starbucks and smell that coffee smell, the smell of the coffee has diffused throughout the coffee shop. You can soak a gummy bear in water and the water will diffuse into the gummy bear and it will get larger. Those are several examples of diffusion.
So yeah, diffusion is just whatever is the lower concentration going into the higher concentration going into the lower. So normally if we were in lab, we'd do an experiment where we would have an egg, we'd put it in vinegar, let it soak, and then all the water would come into the egg and it would actually become bouncy. Like you can drop it on the ground and like it would bounce. So if you want to try that on your own, you definitely can. Don't like throw it on the ground to bounce it because it will pop. But if you just kind of drop it, it'll kind of like bounce. So that's always cool. Vinegar and an egg. So that's diffusion. So now we're going to look at osmosis. So osmosis is the passive, so requiring no energy, the passive transport of water across a selectively permeable membrane to an area of high concentration to low concentration. So we're going to look at the meaning of solvents after this slide, but osmosis is looking at water. That's the important distinguishment to make between osmosis and diffusion, looking at water. So here's a little video that I did of osmosis. So yeah, that's osmosis, water going from high to low concentration. So you can see the red ones are the water molecules and they are, no, I don't know why osmosis and diffusion confuses me, so we're just gonna not talk too much on that. So we're going to look at the difference between a solvent, solute, and solution. So a solvent will dissolve a solute, so you can think of it as water. A solute is the substance being dissolved, so you can think of sugar. And a solution is what forms whenever the solute is dissolved in a solvent, so like simple sugar or simple syrup. So water dissolves it, sugar would be being dissolved, and the solution is what's formed. So you might hear that throughout anatomy. I can't remember, so maybe just kind of understand those. So those are the osmotic components: solvent, solute, solution. Solvent dissolves, solute is being dissolved, and the solution is the combination of the two. So now we're going to look at osmotic pressures. So this is looking at hypertonic, isotonic, and hypotonic solutions. So this is our hypertonic solution. This is our isotonic and hypotonic. So isotonic is even. So we just see even things going here. So whenever we work through these, we're going to think of the example of a red blood cell. So first we start with hypertonic. So if we drop a healthy blood cell into a hypertonic solution, that cell will shrink. So that hypertonic solution, you can think of salt water. So if we drop a healthy red blood cell into salt water, it will shrink because the water inside the cell will go to the area of low concentration in the salt water. Oh, it's so much easier to explain when I have a whiteboard. Sorry, my cat's going crazy. If I could draw this out, it would make a lot of sense. But think of hypertonic solution as like salt water. Another way to remember it is, and how I'll ask y'all this is like, will the cell shrink, will it swell, or will it stay the same? So let me get my pen thing pulled up. So hypertonic, you can think that E is real shriveled or shrunk. So whenever you drop a cell in a hypertonic solution, it will shrink or shrivel. So you can think that E looks real shrunken and shriveled. Where's my pen? And then for hypotonic, doesn't show me where I'm drawing. That O is real swollen. So the cell will swell and burst. So you can think of hypotonic as like distilled water. So if you put a cell into distilled water, the water will move into the cell. So hypotonic, you can think of the O or hypo, hippo. It will swell and burst. Hyper, I don't know why I can't see my mouse. Where is it? There we go. Hyper is just swelling. So again, it would be like dropping a healthy red blood cell into salt water, and the water from the cell would rush out and it would shrink. Hypo would be putting a healthy cell into like distilled water, and the water would rush into it, making it swell and burst. Isotonic is just an even concentration, like the insides of our body. So the pH is balanced, the water is balanced, so the cell stays healthy because there's equal amounts of solute. So just know for this one, because it's really hard to explain without having a whiteboard, just know the shrink and the swell and the even. Okay, so that was it for chapter 2 and 3. We're not going to be doing this. So do I have any questions before we move on to the next PowerPoint? Cool. Alrighty, so let me get the next one pulled up. Screen share, lab 3. So now we're going to be looking at tissues and the skin. So this is going to be chapter 4 and 5. So our objectives for chapter 4, tissues, are going to be looking at the different categories of tissues. We're going to look at the 4 basic tissue types. We're going to look at the location and function of the tissue types, and we are going to look at specific specific epithelial, connective, muscle, and nervous tissue. So this is going to be the start of lab that's no longer review. This is going to be new things for y'all. So you definitely want to pay attention as we get into this. So if we were in lab, this would be an entire microscopic lab. We would be in front of the microscopes, we'd be looking in at cells. It's actually a lot harder to use the microscopes because the cell, it's harder to identify the sections of the cell. So even though we're not having the fun part of looking into microscopes, it's easier to just look at the pictures that we're going to be looking at. So when we look at tissues, we are looking at a combination of similar cells that are grouped together to form a particular function. So we have different tissues that will provide different functions, such as protection, secretion, contraction, and transmission of nerve impulses. So I'm not going to quiz y'all on that definition. I'm not going to test y'all on it. Because it'll start to just make more sense as we go through the PowerPoint. So don't get overwhelmed trying to learn this definition. So whenever we are looking at the study of tissues, we are looking at histology. So normally this lab before we got our new manual was called histology, the study of histology, looking at tissues. So I do want you to know that histology is the study of tissues. Anytime you see the prefix ology, that means the study of. So like gastrointestinology is looking at studying the GI tract. Pulmonology is looking at studying the lungs, things like that. So ology means the study of, hist means tissue. So with the study of tissues, we use a microscope to observe the tissues, but again, we're just going to be using pictures for this lab. So we have 4 basic tissue types. You want to know these. These will be heavily emphasized in lecture and definitely on my exams and quizzes. So the 4 basic tissue types are epithelial, connective, muscle, and nervous. So those are the 4 main tissue types that we see in the body. So let's look at what each of these are, what their function is, and where their location is. So first we have epithelial tissue. Epithelial tissue covers the surface of other tissues, forms the lining of the body cavities, and forms glands. The cells of epithelial tissue are really closely packed together. They have a free end and a basal end. Not super important to know because you'll actually really study those in lecture very, very deeply, but you don't need to know this for my lab. And we identify epithelial tissue based on its size or its shape and the layers present. So I definitely want you to know the function of epithelial tissue. Again, it covers the surface of other tissues. We'll see that it lines our internal organs. It forms the outer covering of our internal organs, and it will form glands. So let's look at it with some real examples. So we have a couple different types of epithelial tissue. We have simple, stratified, and pseudostratified epithelial tissue. So simple epithelium, which if you're talking about just one type, it's called epithelium. Simple epithelium is 1 cell layer thick, so just one. And this would be for easy molecule transport. So whenever we get into the respiratory system, there are these things in our lungs called alveoli. That's where we see the diffusion of oxygen into the cell and carbon dioxide out. And the tissue type we see at the alveoli is simple epithelium because we want that easy transport of oxygen in and carbon dioxide out. So we'll see simple epithelium anywhere in the body where we need easy transport. And again, it's just 1 cell layer thick. Then we have stratified epithelium. This has multiple layers, 1, 2, 3, 4, 5, all the way up to like 14, whatever. So stratified is multiple cell layers thick, and it will provide protection. So we have stratified epithelium in our cheeks. So lining, again, the function is going to line cavities, so it lines the mouth cavity, and it makes it to where if you eat something like a chip, it doesn't pop through the other side of your cheek. It does not puncture through because we have those multiple layers. Another thing about epithelial tissue is it is the easiest tissue, and you don't need to know this, but just so you know, it's the easiest tissue to replace. So our skin is made of epithelial tissue, stratified epithelium. So, you can get a scratch on your skin and it's not like you can see your bone. So it's really easy to replace because we have all these layers and it's constantly shedding and replacing itself. So stratified epithelium, we see that, in the cheek, in the skin, and anywhere where we need some type of protection. Then we have pseudo-stratified epithelium. So this is a single layer, but it looks like multiple layers just because of how the nuclei are arranged. These little purple guys and kind of dark red here are the nuclei. So here it looks like it's multiple layers, but it's only one. So pseudo means false or fake, and stratified, means multiple layers. So fake multiple layers. So again, it looks like multiple, but it's only one. So that's how we base epithelium off of its layers. But we have to add one more segment to define it fully. Now we have to add its shape. So basing it off its shape, we have squamous, cuboidal, and columnar. So squamous epithelium looks flat. You can compare it to like a fried egg or a pancake or a fish scale. It's really flat cell shapes. So like that. Then we have cuboidal, which is cube shaped. The name makes sense, cuboidal, cubed. Looks like a cube. And then we have columnar, which is column shaped. It's long and tall. So, makes sense again, columnar column. So squamous, that flat kind of fried egg, pancake looking cell, and then cuboidal, cubed, columnar column. So let's look at some specific epithelial tissues. So first we have simple squamous epithelial tissue, or simple squamous epithelium. So this is a single layer, we see that from simple, of flat scale-like cells, squamous. So a single layer of flat scale-like cells. Its function will be filtration, diffusion, osmosis, and secretion. So again, we'll see these lining our blood vessels, those alveoli I talked about in the lungs. In our blood vessels, we want, that easy transport of molecules into and out of the blood. So we want to be able to get oxygen into the blood, carbon dioxide out. We want to get supplements such as iron, hemoglobin, things like that into the blood vessels. And we want to be able to get toxins like drugs, urea, things like that out of the blood. So that's where we'll see diffusion and osmosis. And, to do that, we have filtration and secretion. So you're definitely going to want to know the description of simple squamous. The name makes sense if you understand the shapes and the layer component. And then I want you to know that the location, because that's, if you think of the function, that's applying it. So I could ask on your quiz, what cell type, and I try to word the questions on the quiz to be simple, not like really hard. So I would ask what tissue type provides for easy diffusion? Simple squamous, because it's that simple one layer of flat cells, so that easy in and out. Or I might ask, what type of tissue would we see And the, no, because we haven't learned about the blood vessels or alveoli. So you're just definitely going to want to know diffusion. That's the main one that happens here. So definitely know simple squamous is associated with diffusion, easy transport in and out of the cell. If we looked in the microscope, this is what we could see, just that single layer of flat scale-like cells, just like that. So now we're gonna look at stratified squamous epithelium. So stratified, we know, means several layers, and squamous is flat scale-like cells, or flat pancake-looking cells. So we will find this in the epidermis of the skin. The epidermis is the top layer of the skin. When we look at the skin, just a second, it has 3 layers: the epidermis, the dermis, and the hypodermis, or the subcutaneous layer. So this is going to be that top layer of skin. So here's actually a picture of the epidermis, and you can see that several layers of flat cells. And this is because if we get a scratch or a cut, you know, our internal body will not be exposed. So this is providing that protection to the underlying structures. So definitely, you know, the description, you're going to want to know the description for every cell type that we go over today. I want you to know the location, the skin. Stratified squamous forms the skin. And, you know, if you understand that it forms the skin, the function just makes sense: protection. Multiple layers for protection, just like sunscreen. If you put on multiple layers of sunscreen, more protection. And again, if we looked at the microscopes, this is what it would look like. You know, you can see those nuclei, multiple layers of them. So now we're going to look at simple cuboidal, simple one layer cuboidal, cube-like or cube-shaped cells. And what we're going to see is that these line the ducts of numerous glands. So we'll see them in the tubules of the kidneys and secretory portions of small glands. And their function is for secretion and absorption. I don't think I asked any questions on the location or function of this one, so just the description's good. And you don't even need to know, or yeah, I guess you can associate it with ducts. Ducts, not ducks. But yeah, it's going to do secretion and absorption because it's thick. So we'll see, you know, mucus being able to be secreted and stuff from there. So now we have simple columnar, a single layer of column-shaped cells. And what we see is that these are associated with goblet cells. Goblet cells, if you've learned them in lecture so far, are cells that secrete mucus. So again, goblet cells are cells that make and secrete mucus. I do want you to know that. So what we see is that this simple columnar epithelium will line the stomach and the GI tract, and its function is absorption and secretion of mucus. So an example would be a part of our GI tract is our esophagus, what we swallow food down. So whenever you swallow something dry, like let's say you ate a piece of bread or some chips or something, some crackers, we need mucus to get it down. We need that mucus to get it to go down the esophagus. If we didn't have those goblet cells producing mucus, it would have a much higher chance of just getting stuck or scratching and tearing the sides of the esophagus, which again, the esophagus is the tube, which you don't need to know this until we go over the GI tract, but it's that tube that whenever you eat food is gonna go down and connect to the stomach. But again, don't need to know that until we go over digestive. But if you wanna associate that with simple columnar, you can think like, yeah, whenever we eat something, we need that simple columnar to make mucus so we can actually get it down into our stomach. So I do want you to know that it's associated with goblet cells. If you know that it lines the stomach and GI tract, the function is pretty, or I guess the function would be better to know. And if you know the function, then the location's kind of easy. Absorption and secretion of mucus. So this is my favorite one to put on an exam and a practical. Normally I put it on both because if you say you know this to your friends, you sound really freaking smart. So pseudostratified ciliated columnar epithelium. That just sounds really cool to say. So let's break it down. Pseudostratified, false multiple layers. Ciliated, if you remember back to biology, that's going to be hair-like cells sticking off. You can see the cilia right there. Columnar, column-shaped epithelium. So the description says it is one layer giving the impressions of several layers. That's where we get the pseudostratified. The cells are of different heights with nuclei at different levels. So you can see that in this picture. The nuclei look like they're stratified, but they're not. They're falsely stratified, pseudostratified. And it does contain goblet cells via the columnar tissue. Columnar tissue also, we just went over that. And then ciliated, those hair-like cells. So if you didn't write that in your notes real quick, ciliated hair-like cells. So we are going to find these cells in the trachea and the upper respiratory tract, so like our nose. So why we would find them here is because the trachea is what air goes down. The trachea sits in front of the esophagus. You don't need to know that, but just for reference, the trachea is what you can actually touch. That's the trachea. So why it would line the trachea, and again, food doesn't go down the trachea, only air. But let's say you swallowed it down the wrong tube. We want those cilia to be able to push it back up. So those hair-like cells will push whatever back up. If you inhale some water on accident, those ciliated cells are going to push it back up. Let's say, with our nose, if anything goes in our nose, we need those cilia to either push it back. So if you have like a bunch of snot in your nose, those ciliated cells will push it back. And if you inhale something you shouldn't have, such as dust or whatever, it will push it down. So just know those cilia, they make sense, lining the trachea, the nose, the upper respiratory tract. We need to move things there. We also see the goblet cells there because columnar has goblet cells. So of course we make mucus in our nose. Of course, we have mucus lining the throat to kind of make the air not so harsh when it goes down. You need to kind of humidify it and warm it. We'll learn that in respiratory, but that kind of makes sense. So the function of pseudostratified ciliated columnar epithelium is, again, the secretion of mucus and movement of mucus by ciliary action. So this is that picture. You can see those hair-like cells, those false multiple layers, and they're long and column shaped. So for this slide, you might see either a picture where I'm like, identify the tissue type below, or I might say what tissue type lines the trachea. So definitely be able to base this question. You'll see it on the quiz and you'll see it on the practical. So definitely nail this word down because it's a hard word to nail down. Pseudostratified ciliated columnar epithelium. Once you break it down, it makes sense, but it's easy to forget. Some people might put pseudostratified ciliated cuboidal epithelium. So definitely know each section of this word. So that was epithelial tissue. Again, epithelial tissue lines our internal organs and, provide secretion and absorption. So those are the main things: epithelial, lining our organs, lining our body cavities, and doing secretion and absorption. I used to get epithelial tissue really confused with the other tissue types, so just make sure you make that distinction. So now we're gonna look at connective tissue. Connective tissue will all be named based on its location. So you won't see any like simple or cuboidal or anything like that. These all have specific names. So definitely make sure anytime you see simple, stratified, pseudostratified, squamous, cuboidal, columnar, associate that with epithelial tissue. The rest of the tissues we're looking at from here on out are specifically named. So what we see as connective tissue is the most abundant and diverse tissue type in the body. Its function is to provide support, protection, binding, and insulation. So what we'll see is it forms our bones, it forms our blood, it forms our fat cells, it forms all of that. So it is the most abundant and diverse tissue type in the body. What we see when we look at the cells, they're surrounded by a non-living extracellular matrix containing ground substance and fibers. Don't need to know that from my lab at all. You learn that in lecture. I don't remember it that much from lecture, but you will have to know that for your exam one, I believe, in lecture. And what we see is that each individual tissue type is unique. So all bones will have similarities, all cartilages will have similarities, the blood will have similarities. So connective tissue is very diverse. It forms our bones, but it also forms our blood, and it also forms other parts of our body, our adipose tissue. So it's very unique. So for this one, I definitely want you to know it's the most abundant one in the body, and I also want you to know you don't even really need to know the function because it'll just make sense as we go through. So let me make sure my computer is not about to die. Yes, it is. Get it plugged in really quick. Okay. So first we're going to look at mesenchyme tissue. This will not be one I'm going to quiz you or test you over because when I took this lab, it was not taught. The new lab manual teaches it, but I don't really get it and I don't think it's that important. So what it is embryonic connective tissue. So it is the type of tissue we see in the embryonic stages of life. So whenever you're an embryo, you are just made up of these mesenchyme cells. And what it does is give rise to all other types of connective tissue. So it will be what forms all of the other ones. But again, you don't need to know this one. And if we were looking at a microscope, this is what it would look like, but you don't need to know that. So now we have areolar tissue. This is one that I'm going to want you to know. So areolar tissue is what covers muscles, nerves, etc. You definitely don't need to know this for my lab. I don't really remember what fibers and ground substance are, so you don't need to know that. But I do want you to know that it covers muscles, nerves, and etc. Or not and etc. But covers muscles and nerves. So for example, if you cook, which I don't really like to cook, but so I'll buy like a rotisserie chicken. So if you ever buy like a rotisserie chicken or cook some chicken, you know how if you kind of rub it, like some white stuff comes off of it, like some white tissue, that's areolar tissue. So anytime you see that kind of, like if you ever make chicken or buy some rotisserie chicken and you kind of rub it, that white kind of dry stuff that comes off is areolar tissue. And what it does is provide elasticity, strength, and support to connecting tissues and organs. Hang on, my computer just close. Okay, there we go. So if we're in lab, what I'd have us do is kind of just say like, because I will put labeling pictures for these. So you'll either see me ask the location or identify the picture. So does anyone have like an idea of what we could compare this to kind of remember, like, what does this look like to y'all? Like just anything, like anything in the real world. Like what does that kind of look like? Oh, they're so shy. Okay, I'll do the first one, but y'all have to give me examples for the second one. So normally a lot of people say it just looks like strands of hair. So areolar tissue looks like strands of hair. So if I put this on, I think areolar looks like strands of hair. So hang on, close that. So yeah, again, covers muscles, etc. Then we have adipose tissue. This one's super important to know. It is it's where fat is stored. So it makes fat. So it's composed of adipocytes and fat and oil. Adipocytes are fat cells. You don't need to know that. Probably in lecture you do, but not for lab. And again, it's going to be anywhere where fat is stored. We store fat for insulation, energy, protection, support, all that. And so, you know, fat will provide for heat. It will provide for more energy, protection of bones, protection of the eye socket, things like that. So what do y'all think this picture kind of looks like?
I think it looks like clouds.
Clouds, I've definitely heard that one before. So clouds, sometimes people say it looks like bubbles. So you can think of adipose tissue as clouds or bubbles. So if we were looking at a slide, that's what it would look like. Or like that. Adipose tissue is always the hardest one to see because it's very light colored. And throughout lab, it'll normally be depicted in yellow, but you don't need to know. So yeah, adipose tissue, fat tissue. Looks like bubbles or clouds. So now we have reticular tissue. So we will find these in the lymph nodes, the spleen, and the bone marrow. You don't need to know that. I just like the picture of it. So what do y'all think that this looks like?
I'll go again. I think it kind of looks like maybe like blossoms or something. Like
Yeah,
something like
the
that.
cherry blossom trees. Yeah, I always say it looks like cherry blossom trees. So whenever you see reticular tissue, it looks like cherry blossom trees. So definitely know that. Then we have dense regular. This is going to form our tendons and ligaments. So it will serve as a strong attachment between muscles and bones, which is tendons, and bones ligaments. Wait, that might be wrong. Wait until we get to skeletal to make the difference between tendons and ligaments, because that might be flipped around. But again, dense regular is going to be found in the tendons and ligaments. You can see it right there. It kind of looks like what would form tendons and ligaments. I don't think I put that one on the quiz or anything. And the picture, yeah. Okay, so now we have hyaline cartilage. So hyaline cartilage is found in the embryonic skeleton, but most importantly, it covers the end of long bones and joints, and it also forms the nose and the trachea. It forms those because it will provide a smooth surface, enabling movement, and it'll also provide flexibility and support. So your nose is flexible, but it's supportive. You can get like kind of punched in it and it'll be okay. Same with the trachea. You can hit your throat and it's okay. So it's flexible, but it does provide support. It doesn't, just collapse. Our ears are made of elastic cartilage, and that's why they're a lot looser. But the nose and the trachea still need to have support in them. It also covers the end of long bones and joints, which we'll look at more when we get to skeletal and joints, so you don't need to know that. I like the picture of this one, so normally this is the picture I put on. Anyone have an idea? Maybe I can hear from someone else. Don't be shy. Anyone?
Of like what it looks like?
Yeah,
A prickly pear.
that's a good one. I have not heard that one before, but interesting. Anyone else?
I think the top one looks like a lava lamp.
True, a lava lamp. Yeah, I like that one. The ones I hear most commonly are, this one looks like ladybugs or watermelon seeds or fish eyes. I hear those a lot. I always think it looks the most like ladybugs. And then the top one kind of looks like an octopus arm. So, but I like this picture more. I don't use the top one. I use the bottom one because it's more like noticeable. So hyaline cartilage, ladybugs, watermelons, things like that. So now we have elastic cartilage. So the elastic cartilage will form the external ears and the tip of the nose. So it will support and maintain the shape of the ear and the auditory canal. So it's real loose. You can move your nose around a lot and it's really loose. You can move your ears around a lot. It's loose. And here's the picture for it. Any ideas on what this one kind of looks like?
Okay, this is really random, but it reminds me of like cartoon ham, like when you cut it and there's like.
That's one I get every semester. Yes, it looks like ham, like a ham bone inside of ham.
Yeah.
Yeah, I get that one. And then my favorite one when I was, oh, was someone talking? Oh, no. My favorite one when I was my first semester as an IA, I was shadowing under another IA, and she worked in the trauma unit at the hospital. And what she would tell her students what it looks like is whenever they would have someone come in and they would do a cross-section amputation above the knee, and it would look like the bone in the knee and the tissue surrounding it. So I always think of a cut off leg whenever I see that picture. So I like the picture of this one, so you'll probably see a picture for elastic. Now we're gonna look at compact bone tissue. So we're gonna look very heavily at this tissue picture when we get into skeletal. There is actually a whole chapter dedicated to this top picture here, but we'll save that for later. I just want you to know that, of course, it forms our bones. Compact bone tissue forms our bones. It will store minerals, support the body, tissues, and organs, and work with the muscular system for movement. So this one, just if I say what tissue type forms bones, compact bone tissue. You can't just say bone tissue, you've got to say compact bone tissue because there are multiple bone tissues that we'll look at, but compact is the main one. There's also spongy bone tissue, but no, compact is what forms the majority of the bones. So this picture, I always get that it looks like a cut off like tree trunk, so a tree trunk for that picture. So you might see either a picture or what tissue type forms our bones, compact bone. And then lastly, for connective, we're gonna look at the blood. And again, there are so many more types of connective tissue, so many more, but these are the ones that we focus on for lab because they're the most major ones. So blood is a type of connective tissue. A lot of people don't think that blood is a tissue, but it is. So it consists of red blood cells, white blood cells, and platelets. You don't need to know that. We'll study that whenever we get into the cardiovascular system, the vessels, the blood. But we do find it in the blood vessels and in the heart. That's the only locations you find blood in the body, in the vessels and in the heart. Vessels, vascularize all the, sections of our body, like our fingers, our nose, our toes, all of our body has blood flowing through it, but only through blood vessels. And the heart is what pumps it. So we would only find blood in the blood vessels and heart. You don't need to know the functions yet because we'll again look at those later, but red blood cells are responsible for transporting oxygen and removing carbon dioxide. White blood cells are our immune response cells, so they fight allergic reactions, sicknesses, things like that. And then platelets will form blood clots. But yeah, this picture makes sense, but I don't think I put blood tissue on the quiz, so don't stress too much about it. So that was all connective tissue. So connective tissue, all the names are very specific to this, to the tissue type. So again, epithelial has like the breakdown of the name, connective is very specific. Now we're gonna look at muscle tissue or muscular tissue. Again, our part 2, we'll be studying this a lot more when we get to the muscular system and the skeletal system. So we're just gonna go over like the very basic muscle tissue types. So we have 3 types of muscle tissue: skeletal muscle, smooth muscle, and cardiac muscle. We want you to know that. We have skeletal muscle, smooth muscle, and cardiac muscle. So they are specialized tissue for contraction. The whole purpose of these 3 tissues is to provide contractions. That makes sense. Muscles are how, provide contractions. Contracting your arm, lifting your hand, all that's a contraction. Everything you're doing when you're moving your muscles is a contraction. So muscle tissue, of course, would provide for contractions. We see that the cells contain myosin and actin. You don't need to know that for my lab. You'll really study that when you get to lecture. We'll really study it when we get to the muscular system, but for now you don't need to know that. And yeah, do I have? No, I don't have. So I guess there's some different, characteristics of them. I thought we went over them in today's lab, but I guess not. So just know the 3 types: smooth, skeletal, cardiac. Smooth, just for reference, we would find in like the stomach, the places like where you would see epithelial tissue a lot. Smooth muscle provides this wave-like movement called peristalsis. So again, these muscular tissues are providing contractions. Whenever we have food in our stomach, it needs to move into the intestines. So smooth muscle provides this soft wave-like movement to get it down. Skeletal muscle provides strong movement. So it's how we, push something or flex, things like that. And then cardiac tissue is just found in the heart, and it is specialized in ways that we'll learn in the future. You don't need to know those descriptions, but in case you have trouble remembering them, that's how you can kind of associate them. And then lastly, we have nervous tissue, which part 3 will be studied much more when we get to, I don't know why I have skeletal tissue on all 3 of these. That should say nervous system, not skeletal system. But nervous tissue is a highly specialized tissue that conducts nerve impulses. It contains neurons and neuroglia. Neurons are the main cells of the nervous system. We'll look very heavily at them when we get into the nervous system. This is actually a neuron right here, but we will look at this much more when we get to the skeletal, or now I'm saying it, the nervous. So the main things because we're done with tissues now, the main things I want you to take away, and again, I'll reiterate this after we finish the skin, but just for now, epithelial tissue, I want you to be able to identify them based on their layers and their shape. Epithelial, I want you to know, the specific names, like stuff like that. skeletal, just know the 3 types, or sorry, muscular, just know the 3 types, and then nervous, just know it's, you know, found in the nervous system. It conducts nerve impulses. So that was it for histology or tissues. We might have one part of this on the lab report, but I'm not sure. We'll go over that at the end. So now we're gonna get into the integumentary system, or the skin, and then we're gonna finish up. What time is it? 6:30. So I just have like 3-4 slides over this. So this is chapter 5. We've done 1, 2, 3, 4, and now we're on 5. So we're gonna look at the different layers of the skin, and we're going to look at the major parts of the skin. So the skin is divided into 2 regions, but it has a third one. It should say 3 regions. I don't know why it only says 2. But we have the epidermis, which is the top layer or the outer layer or the most superficial layer. It's what makes the, you know, outer layer of the skin. The prefix epi means on top and dermis means skin. So the top layer of the skin, and it contains keratin. You might have heard of like shampoos that contain keratin. Keratin is this protein that will take over the cell and make it waterproof and strong. So keratin is a protein that will strengthen the cell. So for example, ducks, like the animal ducks, have a lot of keratin in their feathers. So whenever they go into the water, you know, the water comes off of them real quickly. They don't stay wet for a long time. So we see keratin in feathers. You can get keratin supplements for your hair, which can help with like frizziness, I think, because if it's waterproof, it probably won't get as frizzy. And it'll strengthen the hair. So we see keratin in the top layer of the skin to strengthen it and make it waterproof. Whenever you put your hand in a bowl of water, it doesn't, you know, soak in unless you leave it in there for a long time, and then your hands get all crinkly. But if you just dip it in, you can just flick the water right off. It didn't like make water come out or anything like that. Then we have the dermis, which is the middle layer of the skin. It has connective tissue that supports glands, and then hair, sweat glands, etc. You know, for the layers of the skin, I might have you identify a picture. So definitely know epidermis is that top, dermis is that middle, and then hypodermis is the bottom layer. It's also called the subcutaneous layer. That's how I learned it, but I'll always use hypodermis / subcutaneous just in case you learn it one way or the other. I'll put both. Hypo means below, so below the dermis. So epidermis, dermis, hypodermis. The hypodermis contains adipose tissue, so fat. The dermis contains like our glands for our hair, our oil glands, our sweat glands, and then the epidermis contains keratin and sheds a lot. But you'll study the skin much more in depth in lecture. So for me, I just want you to know, be able to label, you know, a picture of the skin: epidermis, dermis, hypodermis. Or I might say like, what's the most superficial layer of the skin? The epidermis. Or what's the top layer of the skin? What's the middle layer of the skin? What's the innermost layer of the skin? What's the most deep layer of the skin? So it makes sense if you just break the words down in your head for a second. Some of the other IAs will take this picture and definitely look a lot more deeper into it, look at the hair follicle, look at the sweat glands, what forms the sweat glands, things like that. But we're exercise and sports science majors. I'll leave that for like dermatologists. So now we'll look at the nails. So the nails are of course located at the distal end of the fingers or the digits. So they're the distal or furthest most part away. Their function is to protect the fingers and grip and manipulate objects. So, you know, if you hit your finger on the nail, it doesn't hurt, but if you hit it on the bone, it kind of hurts. So the nail will kind of provide that protection to the end of our fingers, because that's what we use the most. So what we see is that it has a free edge, a body, and a root, and it also has a cuticle. So let's look at this picture. The free edge is just, the free edge of the nail, the part that, kind of sticks off a little bit. Then we have the body of the nail, so the body, the middle part. And then we have the lanula, which is that little white part that you can kind of see at the bottom that sticks up a little bit. You can see it a lot more if you put pressure on your finger. So if you kind of squeeze it hard, you can see the lanula a little bit more. And then we have I can never say it, epiconichium. I don't know how you pronounce that. Epiconichium. I looked it up last year, but I can't remember now. Eponychium, I'm not sure, but that's the cuticle. So that, part that kind of at the nail salons, if you get your nails done, they cut it off, and it's really like scary feeling, and I just can't get my nails done because of that. So I would definitely be able to label this picture, but only know the free edge, the body, the lanula, and the epicuniculum, whatever, how you say that. You don't need to know like the lateral fold or anything like that. So that's the nails. And normally we'd look at sensory physiology, but I'll save this for the nervous system. I'm not even going to go over it right now. So I'm going to take y'all through what I want you to know for the quiz. I will walk y'all through the lab report, and then we will be done. So I accidentally closed out the other PowerPoint. So let me just do the preview on Canvas real quick. So for cells and mitosis, plasma membrane, pretty basic. Y'all know it. Cytosol, just that fluid inside the cell. Nucleus, control center of the cell, contains DNA. Mitochondria, powerhouse of the cell, turns nutrients into energy or ATP. The phases of mitosis in order, so PMAT, PMAT, prophase, metaphase, anaphase, telophase. Be able to distinguish passive transport from active transport. Passive transport requires energy, passive transport does not require energy. Active transport does require energy. Just know diffusion is, transport of molecules from high to low. We all know that, high to low. Osmosis is transport of water from high to low. I don't even think I'm gonna put these on there. Definitely know hypertonic, isotonic, and hypotonic in relation to a red blood cell. So if I drop a red blood cell in a hypertonic solution, what will happen to it? It will shrink. So definitely know those three in relation to red blood cells. So that's it from the first part of lab, and then the second part of lab. Load for some reason, it wouldn't load for me the other week. Okay, I'm just gonna go back to the PowerPoint because that's gonna take forever. So share PowerPoint. So for this PowerPoint, know histology is the study of tissues. Know the 4 tissue types: epithelial, connective, muscle, and nervous tissue. know the basic functions of each of the tissue types. Function of epithelial, again, covers tissues, forms the lining of cavities and glands. You're going to want to know the distinction between all the layers and all the shapes. So you need to understand each layer and each shape. And then you're going to want to know, the epithelial tissue types. Simple squamous makes sense. Easy transport diffusion. Stratified squamous, multiple layers, flat, the skin. Simple cuboidal, single layer, cube-like. Simple columnar, contains goblet cells which form mucus and lines the stomach and GI tract. Pseudostratified ciliated columnar, my absolute favorite one. We went over these already, so know what I talked about and, be able to identify a picture of this one. No, connective tissue is the most abundant tissue type. We don't need to know mesenchyme, don't need to know that one. Areolar, I want you to know that one. Adipose, I want you to know that one. Reticular, I want you to be able to identify the picture. Picture for hyaline. I just hate tissues. They're my least favorite part of this lab. Elastic, picture, maybe ears, tip of nose. Compact bone, forms bone, know the picture. We're not gonna do blood. Know the 3 types of muscular tissue, smooth, skeletal, cardiac. Again, smooth, skeletal, cardiac. Nervous tissue, I'm not gonna ask you any questions on it, but just know that that's one of the 4 tissue types. Skin, what I just said, know the 3 layers, know which one's the top, the innermost or middle, and the bottom. Know that the epidermis has keratin, and then be able to label this picture of the nails. And I know I tell y'all all that, and of course you won't see every single one of those things I tell you on the quiz, but you still do need to know them for future classes. So those are what I want you to study. Of course you won't see every single one of those on the quiz, but those are what you need to know for lecture, for life in general. So let me pull us back up to Canvas just to kind of close out for the day. Sorry, this one went a little bit longer because we had to fit in last week's second part, but whatever. So here's the supplemental readings you can do if you are a little confused. They honestly really do help. And then your lab report is kind of a little bit more fun this week, not super fun. So what I'm gonna have y'all do is make a flow chart of the order of mitosis. So you can just, do prophase, line, metaphase, line, anaphase, line, telophase, line. You can draw it out on a piece of paper, or you can do it on like Microsoft Word or whatever. And then I want you to draw the different shapes and layers of epithelial tissue. So you can literally just draw exactly what I put on the slide, draw the simple shape, stratified, draw the multiples, pseudostratified, draw how it is, and then draw squamous, cuboidal, and columnar. Those are super easy. You don't have to I'm not good at drawing. I wish I was. So you don't have to be an artist or anything. I had students during the summer that had computer-based drawing programs. So if you want to draw it on a computer and take a screenshot, if you want to draw it on Snapchat and then take a screenshot, whatever, just find a way to draw it or on paper. Then I want you to draw the connective tissues. If we were doing homework out of the lab book, you would have to do a lot more drawing for this lab. So I know it seems like a lot, but it's really not. So draw the connective tissues, areolar, just draw like things that look like hair, adipose, bubbles, or clouds, reticular, the cherry blossom trees, dense regular, the waves. hyaline, the ham bone, or wait, hyaline is the ladybugs, elastic, ham bone, compact bone, the tree trunk. So just draw, and you can literally just draw it like in a space that big. You don't have to do a huge piece of paper. You can just make little boxes on a piece of paper and draw them out. And then you just attach your outline. There's no, I know I put 250 word minimum, but there's no word count for this one because you're doing pictures. The other IAs are still doing a word count. They're still doing an essay in a specific format, which I do not think is fun. So just attach your outline. I have y'all do this just so whenever you start studying for your practical, you can go to your assignments and it's all there organized for you. Plus just having to do an outline is a really good way to study. So do I have any questions on the lab report or anything else about lab today?
Did the mitosis flowchart, did that have to have pictures too, or just?
No, just the words. So just draw like prophase line, anaphase line, or metaphase, anaphase, telophase. Cool, cool. Anyone else?
Outline, you just mean like the order of like our pictures?
For the outline, just write like the important, topics we learned today. So
Gotcha.
just, like just sum it all together, like that.
Gotcha.
Anyone? Okay, cool. Well, first of all, I really appreciate y'all helping each other out in the group me. I am so late on all my other classes right now, so I'm trying to get to a point where I have more time to respond and stuff like that, but y'all have been awesome so far with helping each other out. I really appreciate it. And again, no lab next week, so the lab report isn't due for like 2 weeks from yesterday, and the quiz isn't for 2 weeks from today, but definitely, stay on top of material. So it's great seeing y'all. You can log off. I'll stay on in case anyone has questions still. So peace.
Okay, so I completely messed up the date and I didn't do lab report 1. Are we going to have any extra credit opportunities?
So I'm actually waiving lab report 1 if no one got it turned in because we had a Canvas malfunction. So you are totally good there.
Cool. And then, no, actually that's it. Well, thank you.
Sure.
Oh, wait, no, advice. Okay, so, because I'm trying to like stay ahead on top of my things, right? So I've been doing outlines before lecture, like for regular class, and a lot of the stuff that you're covering is what we're going on in lecture. So like, do you think it's good to like write them down in 2 separate journals, or should I just continuously like add it all into one?
Keeping 2 separate journals is really good. Like I had 2 binders for each lab when I took it because lab is anatomy based and lecture is physiology based. So lab we're looking at structures, lecture you're looking at physiology. It's great to have them like side by side. Definitely separate the 2.
Okay, cool. Yeah, because I'm like flipping back and forth, and now I have like a lab, and then lecture, lab. Okay.
Yeah, for sure. And I mean, we try to keep it together, but there will be points where your lecture will kind of fall behind in lab. But it's definitely great to keep them together. And I love that you're doing outlines before. That's how I study. So good job.
Yeah. Trying to keep the healthy habits because already I feel like behind in school. So, but thank you. Keep going. You're awesome.
Thank
Have a
you.
good day.
So this might be a dumb question because I'm not good at math, but like for the quiz, it says like there's 10 questions, right? And each one is 0.5 points.
Oh my gosh, that was supposed to update. Each one is 1 point.
Okay. I was like, I only got 1 question wrong, but it says that like I got less than a 50, so I wanted to like clarify.
Also for that 1 question, which crap, I meant to say that, I have to go in and manually grade the fill in the blank one. So that might be the question that you got wrong, but whenever I manually grade it, you'll get it right. So you might have just like had like a different spelling than how I spelled it, so you might have not even gotten one wrong.
Well, it's just that it says like it says that each question is 0.5 instead of 1. So it says like I didn't get all my points. So I was just trying to make sure that.
Yeah, so that should say a 9 out of 10. I need to go edit that. I'm sorry, I thought I did that yesterday, but
Okay.
thank you for letting me know, because that's definitely good to know. So
Okay. Thank you.
oh, for sure. Bye, girl.